Okay. So muna, actually ang alternative activity that I was thinking instead of this lecture is a talk show type. But when I checked our schedule, I realized that we only have around two meetings before our first long exam. Now, can you still remember sa ating first long exam? I presented it during our course introduction. You forgot already or you don't want to think about it? You don't want to think about your long exam? So we scheduled it on April 26. Okay. So the alternative activity actually is parang summary ng ng lectures kasi ang haba ng lectures, 'di ba? We have 3 lectures in module 2 and we have medyo short lang lectures sa module 3 but we have a lot of videos. All right. So before we start, do you have any question? Do you have questions that you would like me to answer?
I have a question.
Yes, go ahead.
Regarding sa learning resources, kayo nag-check ko ma'am like damo-damo siya or
Oo.
sa exam ma'am should we focus only to the PPT provided or may mga included din sa other resources na ginhatag mo ma'am?
Based on our previous exams sa Fish 154 exams, we just focus on the PowerPoint. And then, di ba may mga terminologies sa PowerPoint that siyempre kung sa PowerPoint daw outline lang siya, daw guide lang siya, no? You have to do further study or research para for you to understand the concepts. Like for example, when we say gram-positive bacteria, di ba, usually ang question would be, it will be compared with gram-negative bacteria. So maybe we'll be asked why is it that gram-negative bacteria cannot retain the crystal violet dye or something like that. Nabudlayan na ka mo sa sample question. Hindi. Pero amo na. So you have to do additional reading on that particular concept para mas ma-justify mo if i-ask us questions like that. So you just have to, hindi ko man gusto bihang balon na you limit sa PowerPoint, pero very useful ang mga other materials. For ang mga journal articles, if may ara gin-attach dara sa module. By the way, class, I am not part of the team who developed the course packs for Fish 154 course, no? But I have handled the course for several SEMs, so I am also familiar with the PowerPoint mismo. But the additional or supplemental materials were provided by the team who developed the course pack. So if they included mga articles or mga laba na bala nga research studies, no, you don't have to spend much time or a lot of your time with that articles, no, because ang purpose lang is to showcase or to show to you the recent advancements when regards to certain topics in Fish 154 or in microbiology in general. So basta mga laba na gid nga mga references, pangitaon nyo lang ang daw mga key concepts or lantawon yung iya abstract na muna lang. Pero for videos, you need to watch all of those videos kay I don't know if it's also useful for you. Pero for me, medyo ano ako, I learn more if I see things visually. Kung ano na bala, medyo visual ako na person. So mas nag-learn ako if makita ko ang interaction, if I can picture out, or if I've seen in a video how a bacteria divides. So mas ma-picture out ko siya doon muna. But your PowerPoint would be enough naman and very comprehensive subong inyong PowerPoint. Parang it's already a standalone material for Fish 154 ang inyong nga PowerPoint lectures. No, if you would notice, medyo damo ang text sa PowerPoint, di ba? Do you agree? Damo text. Because as much as possible, we don't want to super outline lang the content. So medyo detalye ang PowerPoint ninyo. But gina-encourage ka mo to do further reading on the matter or the topic being discussed in the PowerPoint.
Okay, ma'am. Thank you, ma'am. Ano lang kasi, ma'am, ng overwhelming nung pag-chat ko ng Drive.
Oo. Yeah, I understand. Kay ang mga iban gani nga materials hindi ko man ma-upload sa LMS. That's why ginhatag ko na lang gid ang link sa Drive. No, so everybody can access the Drive. Is there any of you who had problems sa Drive? Sa pag-access ng Drive? Wala man. Actually, for today, we will not really discuss per slide, no? We'll just run through the lectures and we will medyo i-elaborate ko lang ang some topics that needs further discussion. So, gaya ng example ko, we need to discuss further the the cell wall of gram-positive and gram-negative bacteria. No, because if ang nakagraduate ka sa if you have taken Fisheries 154, you're supposed to know the difference of gram-positive and gram-negative bacterial cell wall. Okay, so medyo na siya, requirement lang sa mga teachers, no. Okay, so is there any other may questions or concerns from you guys? May pabangkot pa ka mo? Wala man. Okay, sige, screenshot ko na yung aton attendance. So wala na ibang question before we proceed?
Na siguro, ma'am.
Okay. Sige. Sige, ako ang ma-ask. What do you prefer? A discussion like this or a recorded lecture? Anybody? Hindi nyo gusto both?
Lang sa kunya, ma'am, ang ma'am, discussion.
Discussion. Nga, nag-meet online.
Ma'am. Pero kung recorded lecture, ma'am, baka mas more detailed siya.
Actually, on the part of the teacher, no, mas detailed and mas maka-prepare ka gid be kung recorded lecture, no, because if hindi mo like ang imo nga pag-deliver or you missed out some information, you can just repeat the recording, no. However, it's quite time consuming man on our part because, for example, for a lecture, we have to repeat it several times. Kay in my experience, may ara ko class na I really have to do online or I really have to give the students recorded videos or recorded lectures. No, I am spending a lot of time because there are times na ang Zoom ko may audio, minsan wala. So I have to do it like several times. Mga ka 5 times ko siya iliwaton. But good thing about recorded videos is or lectures is you can just replay, no, the lecture if you wanna study. Okay, but well, if you want a recorded video, pwede ko man, or recorded lecture gali, pwede ko man ihatag sa inyo. Okay? Sige, so let me share my screen. Yan ang good class. Oops. May nabatian ka mo nga noise? Wala man. Okay lang.
Wala. Okay
Okay
lang.
lang, okay. Okay. I am working from home and my folks here are listening to the radio. Okay, and I can't just tell them to please turn it off. Okay. Sige. So I added some slides no in this topic, no. Module 2, meet the microbes, the bacteria. Okay, so there's a tagline here, no, or a line which says happiness and bacteria have one thing in common. They multiply by dividing. Very nice ang line, no? So, can you relate with this one? Who can elaborate this before we go into the real micro topic? Happiness and bacteria daw have one thing in common. They multiply by dividing. So your happiness will multiply if you share it. Parang ganun. Parang that's how I interpret it. Anyway, I have a trivia to add to that, no? Have you heard about serotonin? Serotonin is a major chemical involved in the regulation of mood and emotion. Ayan. So, paano siya i-relate sa micro? There was a study conducted by Clark and his team in 2012, which showed that the absence of bacteria, no, during the early stage of life affected the concentration of serotonin during adulthood. So, di ba, serotonin is the one that regulates your mood and emotion. And if affected ang concentration during adulthood, maybe amo nang iban nga dasig sila ma-depress or easily affected sila by the negative vibes around them, no? So the study of Clark proved that or suggested that administration of antibiotics during childhood can lessen or reduce the amount of serotonin during childhood. Ayan. So, do you like the study of Clark? So that's something new, right? Or balaan nyo na. You know, one thing na hindi ko gusto sa online class is medyo mahina ang interaction between the teacher and the students. Right? So if you want to react or comment or whatever, just press the heart or the, I don't know, mga emojis dira sa aton nga Zoom. No, so para somehow medyo naga-interact man kamo. But feel free to speak up or raise your hand if you want to say something. Okay, so that's for today, serotonin ang aton nga bag-o nga na-learn. All right. So we will be studying about bacteria, no? And we'll be talking briefly about their structure, morphology, their modes of reproduction, the spore formation, and their differentiation based on shape, nutrition, oxygen, temperature, and pH requirement. All right, so this is the typical bacterial cell. Ay, gamay ba ang makitaan niyo sa screen? Class?
Wala, naka-full screen.
Gawa, ma'am.
Ang
Dali
pipi.
lang, dali.
Wala siya noon.
Dali lang, gid, ha? Dali lang, gid, class, ha? Because I am using two laptops. Kaya ang isa ko ara tanan ko nga files, but there's no I can't open my Zoom sa isa ko ka unit. Sa isa ko naman ka unit maka-open ko Zoom but guba iya keyboard. I'm so poor. Ayan, sige. Dali lang gid, ha? Okay, sorry. Sorry about that. All right. So what are the external structures of the bacteria that you can remember upon studying Module 2 PowerPoint lectures? Okay, so we have the glycocalyx, or you can also pronounce it as calyx. Okay, so this is external to the cell wall, no? And it is composed of polysaccharide and polypeptide. Ayan. So, it exists in two forms. Okay, so if it is organized and firmly attached to the cell wall, it is called the capsule. And if it is unorganized and loosely attached, it is called the slime layer. Okay, so what is the function of this glycocalyx? So it serves as a source of nutrition of the cell and provides protection against dehydration. So as I have mentioned earlier, no, we will not really read each or every word in the PowerPoint. We will only discuss those important concepts. Ay, wait lang. May notes dire. Paano ba nga wala sang notes?
I-presenter's view lang, ma'am, yung PowerPoint. Naka-presenter's view yung PowerPoint, ma'am.
Play from current slide. Ayan. So wala man siya ano no? Dako nang makita ninyo.
Yes, ma'am. Okay na, ma'am.
Ayan, so that's glycocalyx. Okay. So there are large capsules or small capsules, and there are non-capsulated bacterial cell. Okay. So another external structure is the flagella. So are you familiar with flagella? Sa din siya ginagamit?
For movement, ma'am.
Yes, very good. So during our high school, we were taught that flagella are used by organisms, microorganisms, to propel or to move. So there are 4 arrangements of flagella. So the first one is the monotrichus. No, it only has one flagella, Lophotrichus. Medyo ano siya, parang naka-ponytail sa one end or tuft at one end of the cell. The amphitrichus tuft at both ends of the cell, and the peritrichus are flagella that can be found around the bacterial cell or anywhere in the bacterial cell. So these are the example of organisms that have different arrangements of flagella. Okay, so flagella is composed of 3 major parts. No, the filament. This is the filament. No, it's the most exterior or the exterior part of the flagella, and then we have the hook, the one that's holding the filament and the basal body. No, and of course, we have the basal body here. So para siyang screw that attaches, the basal body serves as the screw, no? Parang lock of your flagella doon sa interior of the cell. Okay, so notice the difference of the flagella of the 2 types of cell, the gram-negative, this one, and the gram-positive bacterium. So for gram-positive bacterium, this one, it only has thinner or ano lang siya, 2 hooks in its basal body, and while this gram-negative bacterium has several, no, sa iya basal body. So what is the idea of this, no? Why is it that may amo nga difference sa flagellar structure of the 2 types of bacterium? It's because the gram-negative bacterium has what? Double or the difference is being explained by the difference in the components of their cell wall. So the plasma membrane of the gram-negative bacterium has interior plasma membrane, and it also has outer membrane, which we will discuss in more detail in the next slides when we discuss the gram staining procedure. Okay. So this slide explains how the microorganism or bacteria move using its flagella. Okay, so the movement is by rotating the basal body. So this is the basal body. So pag in-rotate siya, amo na siya ang nag-manifest sa movement of the whole flagella. So this one, the basal body, is the one that's responsible in the movement of the whole flagella. All right. Yan. So for counterclockwise motion, it makes the bacteria run or swim. The counterclockwise rotation changes the direction of the bacteria in random motion or random direction. And the motor periodically reverses, causing a random change in the direction. So that's how the bacteria moves. So mas namin siya kung we have a video, no? And this one. Ayan, so taxis naman refers to the movement of bacteria towards or away stimuli. So may we have the chemotaxis or the phototaxis. So their name, the naming implies, no, kung chemical ang stimuli, it's chemotaxis. If the stimuli is light, it's phototaxis. Okay. Another external structure is the eggshell filaments. No, it allows bacteria to move in a ano siya, corkscrew motion. Parang have you seen I don't know if you're familiar with the tool used by the carpenters in making holes sa wall. Ano siya, barina ang tawag sa Iloilo. So it moves in a corkscrew motion until it creates a hole sa wood, no? So that's how eggshell filaments work in spirochetes, right? Okay, another external structures or the two common external structures in bacteria are the fimbriae and pili. I actually want to cover some parts of the slide because I want to ask you which one is longer and which one is shorter. So shorter si fimbriae, no? Pili is consist of protein called pilin, which or where it got its name, no? Pilin. Okay, and it is used for attachment rather than motility. Hindi siya for movement ang pilay. Okay, you will learn later that there is one particular mode of reproduction in bacteria that is made possible because of the pili. Okay, so the shorter strands again are the fimbriae. They are longer. They are shorter versus pili, no. But most its purpose is more on locomotion or mobility. Okay, the cell wall. Okay, so cell envelope. By the way, so what are the external structures that you can remember again, just to make sure that you have retained the discussion on external structures before we go into cell envelope?
Flagellum, pili.
Fimbriae.
Pili,
Fimbriae.
fimbriae. Wag ko lagi. Mga last. Okay, what else?
Glycocalyx.
Okay, gycocalyx or gycocalyx. What else?
Eggshell filament.
The eggshell filament. And wala nyo giging panumdum si flagella or may nag-mention, wala lang ko kabati.
Mayara, ma'am. First.
May nag-mention.
Number lang.
Okay. Sige. So we'll now go to cell envelope. All free living bacteria have a cell wall. No, these are the important things that you should note. By the way, can you see my cursor? I'm moving it. Makita nyo?
Yes, ma'am.
Yes, ma'am.
Okay,
Yes,
okay.
ma'am.
So sometimes I'm using it to direct you kung din ako nag-read, okay? So periplasmic space and outer membrane are found in gram-negative bacteria, and the capsule is only found in some strains. May mga specific strains that contains that are capable of producing capsules. Wait, we have a chat. Okay, inubos ni Julian. Okay, very good. Okay, so notice the difference of gram-positive and gram-negative bacteria. So, medyo parang cartoon na pag-prepare ng figure. So, let's go to the next slide. Alam si Ma'am. Okay, so the cell wall is complex, no, semi-rigid structure responsible for the shape of the cell. I'm sure you have heard this about this already in your high school, no, and composed of peptidoglycan, called a peptidoglycan, meaning it is composed of proteoglycan or murine. Okay, so it has long chains of polysaccharide cross-linked by short peptides. So ayan, we will not read all of this because you can read it. You can read this yourselves. Okay, so let's proceed with the cell wall and gram staining. Actually, the reason or something that triggered me to really give a lecture on this, no, is this one, the cell wall of the bacteria. Because, yun, as I've mentioned kanina, it's vital that you know for you to know the components of the cell wall and the difference of the gram positive and gram negative bacteria. All right, so gram positive bacteria consists of many layers of peptidoglycan. Look at this one. So this represents the peptidoglycan layer, no, which is anchored or parang na-hook, no, ng tectoic acid. Teichoic acid. All right. And both have, sorry. Ayan. So this is the gram positive bacteria. It has a periplasmic space here. And then these are the teichoic acid. And it also has plasma membrane on this side. Remove ko ito because, ayan, istorbo. Look at the gram negative cell wall or the cell wall of the gram negative bacterium. It has this outer membrane and then the periplasmic space, no, with this peptidoglycan here. And then it has another plasma membrane on this side. All right. So, so the peptidoglycan layer, just an important things to note, no, is the outer membrane of the cell wall, and its outer surface is covered with lipopolysaccharides. As the name implies, it is consist of sugars, no, linked to lipid membrane or to membrane lipids. All right, so ito yung lipopolysaccharides. Itong mga pink granules on this side. Ayan. Okay, so busin na natin siya. Sorry. So have you heard about gram staining or gram stain technique? Have you heard about gram stain technique, class? Have you done gram
Yes,
staining
ma'am.
in your lab? Sa
Yes.
ibang section,
heard
not
lang,
under
ma'am.
me.
Heard pa lang, ma'am.
Ha?
Heard pa lang kag nakakita sing slide nga gram positive na organ
Saan
nga
ka mo
slide.
nakakita?
Biolab, Bio 14.
Sa ano, ma'am?
Okay. Okay.
Ang F10 sang last sem, sang face to face.
Okay. Sa face to face.
101.
You know what, class? If this 154 course is what? Delivered in a face-to-face mode, you will really appreciate the world of bacteria. Kaya daw ka sa jagged sang sadya sang lab sang 154. Well, sige lang. Hopefully matapos ni tanan base may ara kita nga daw remedial laboratory activity for this course, no? Tani lang may ara. Pero sa jagged niya ang 154 nga lab. Anyway, sige, hindi ta na lang ako pag ipainggiton. Kay wala magkatanan sang mahimo, no? Okay, so the gram stain technique no, is based on the ability of microorganisms to retain the color of the stains used during the process or during the gram stain reaction. And this technique was developed by Hans Christian Gram. Okay, and his publication came out in 1884. So this is actually a very important preliminary step in the initial characterization and classification of bacteria, no? Which is a key procedure in the, yun nga, identification of the bacteria, lalo na sa mga laboratory, no? Kasi kung gusto mong i-identify what causes the disease or illness, no, you can already, by using a gram stain technique, you can already classify or identify the organism based on its major classification, kung gram positive ba siya or gram negative. Ayun. So note that gram positive bacteria stains purple, while gram negative stains red. So, ayun, sinabi ko na to. Initial characterization, classification, or identification and screening of microorganism. Bakit pareho yung picture? Ayan. Like, i-move ko ito. Okay. So, this slide shows the image of gram-positive bacterial cell wall. All right. So it contains an LTA, lipoteichoic acid, which consists of teichoic acids, which act as a regulator of autolytic cell wall enzymes. Diba, I've mentioned earlier about teichoic acid, no, na parang sila yung nag-hook doon sa cell wall para ma-maintain yung integrity ng wall. But this LPA acts as regulator, or parang when the cell is injured or nearing death, pag malapit na siyang mamatay yung cell, pag alam ng cell na malapit na siyang mamatay, this LPA will activate the uramidases, or yung enzymes, autolytic enzyme that will cause the death of the cell. So, yung mga cell kasi, pag feeling nila they can no longer survive, mas okay pa na, okay, I will autolyze na. So, parang ganun. Ayan. So, the peptidoglycan layer is around 20 to 80 nanometers thick, and it has polysaccharide composed of two subunits called the N-acetyl glucosamine and N-acetyl muramic acid. And the layers form, as layers form, they are cross-linked by short chains of polypeptides. No? Actually, binasa ko lang yung sa side ng slide. No? So I have already mentioned about the LTA. So this thick layer of peptidoglycan cell wall shows evidence, no, or indicates the ability of the cell wall to resist decoloration or not, is based on to resist the decoloration process during gram staining. So later we will discuss the gram staining technique or the process. Tapos identify what will happen to gram positive cell wall, what will happen to gram negative cell wall. All right. Ayan. So this one naman, this slide shows the cell wall of gram negative bacterium. So notice the outermost layer, which is the lipopolysaccharide, no, this one, that may become toxic, no, when released during infection, itong lipopolysaccharide, and may function as receptors and blocking immune response. It also contains purines. Okay. Note that gram positive or gram negative bacteria have a thinner thinner layer of peptidoglycan. It's only around 10% of the cell wall. And since it is thinner, it loses the dye, no, that's being used during the process, gram staining process, when added with a decolorizing agent. So, but it retains the counter stain. Later on, you will know what are the counter stain, what is crystal violet, no? And it has gram-negative bacteria has an outer membrane which contains lipids, no, which is separated from the cell wall by means of a periplasmic space. So amo ni siya ang, so this one is a periplasmic space. Okay. Okay. So look at this. This shows the process or the of gram staining technique. All right. So we have here the gram-positive bacteria. We have here the gram-negative bacteria. The end product is the gram-positive will be stained purple, and the gram-negative will stain red or pink. Okay. Again, which one has a thicker peptidoglycan layer? Gram-positive or
Gram
gram-negative?
positive. Gram positive.
Okay, gram-positive. Thank you for answering. Okay, gram staining tests the bacterial cell wall's ability to retain the crystal violet. So since ito ang crystal violet, most likely, kaya siya tinawag na positive kasi nare-retain niya itong crystal violet. Right. So iodine solution produces purple-colored iodine dye complexes. So the process actually of gram staining is, may I ask you again, may nag-gram stain na sa inyo sa ibang section sa lab? May nag-ara na?
Wala pa kami, ma'am.
Nan ka mo under sa akon. Because the section under me is still on activity. Wala pa, next pa namon ang gram staining. Ara pa lang kami sa microbial growth. Microbial growth curve. Nag-ara na kamo sa sa staining?
Wala pa, ma'am.
Okay, so sige, appear. May-ari man din sa inyo na estudyante ko sa lab, no?
Yes, ma'am.
Okay, hi. Okay, so the process is actually you prepare a smear, no, bacterial smear. So how do you do that? You put, teka, i-on ko nga, wala gali naka-on ang ako na video. Sige. Sige, i-off ta lang liwat later kay ano siya, feeling ko gahina ako na connection. The process of gram staining includes bacterial smear preparation. So how do you prepare your smear? So I don't have props here. So for example, this is your slide. You put small amount of water at the center of your microscope slide, and then you spread it using your sterile loop or hook. All right, and then you transfer or you get small few cells of your colony, na bacterial colony. So ibutang mo na dira sa mga water sa center ng slide, and then you hold your slide this way, and then may flame ka na dira, for example, you pass your bacterial water mixture on top of the flame. So over the flame, yamuamo mo na siya until nga mag dry out ang imo nga water. But before you do that, gali, you have to, di ba may water, may bacteria, ipa-air dry mo siya danay. Medyo nag-dry lang siya gamay, saka mo siya amu-amuha na sa flame. Make sure that you don't burn or hindi mo gusto mas-scorch, masunog ang imo nga bacterial cell. So dapat tama-tama lang. The purpose of that is to allow the bacterial cells to be fixed or mag-stick sa imo nga slide. So that's what we call fixation. You have to practice aseptic technique all the time, okay? So once you have that, you add crystal violet solution. So of course, the crystal violet colors, color siya, violet. Alright, so you add that sa imo nga cells, bacterial cells, but your bacterial cells appear colorless. Okay, sa start ng pag-fixation pa lang, it's still colorless. Upon addition of the crystal violet, both the gram-negative and the gram-positive bacterium will stain purple. Okay? Then after that, you add the iodine. Okay. So why do you add the iodine? What is the purpose of iodine? It's a mordant, no? It's called a mordant, M-O-R-D-A-N. Okay? It serves as a mordant. Pag inambal mo siyang mordant, anong definition? Can any of you give the meaning of a mordant? Okay. It's the one that fixes the dye, no? Kumaga daw, it makes your dye stay further sa cell wall. So it makes or it forms a crystal violet iodine complex sa cell wall. All right. So what happens next? After addition sang iodine, may ara nakita sang crystal violet mixture, no, sa or complex nga na form. When it becomes a complex, 'di ba medyo bigger na siya pag complex na kay duha na ka dye, no? You add your ethyl alcohol, which acts naman as a decolorizer, no? Or kung magama ka decolorizer, it will remove the color of the previously added dye sa imo nga cell, no? However, it will only dissolve or it will only decolorize the gram-negative bacteria. Lang tawa dire sa aton nga figure. No, it will decolorize the gram-negative bacteria. Who can explain the reason or the principle behind the decoloration of gram-negative bacteria? Nga naga-decolorize ang gram-negative bacteria?
Kaya na-dissolve, ma'am, ang iyang lipid layer. Charot.
Yeah. Okay na, Mayo, na nga nagasabat ka. Yes, because 'di ba na-notice nyo kagina, let's go back to the structure. Ayan, mas namin igali nga slide oh. Okay, mas makita natin. Pero dito yung answer sa previous slide. Okay, this is the lipid layer, right? So in gram-negative bacteria, after addition of the alcohol, 'di ba, ang lipid ma-dissolve sa alcohol.
Yes
Yung
po.
chem, 'di ba? So pag na-dissolve siya sa alcohol, ari dang imo nga di-iodine, crystal violet iodine complex. So kung na-dissolve na ni siya, wala na ni siya, magwa karon ng imo nga complex. Okay? That explains why naga-decolorize na dula na ang purple because nagwa na ang imo nga crystal violet iodine complex. Whereas sa gram-positive naman, anong hambal sa gram-positive? What happens to the gram-positive bacterial cell? Ara man to sa slide. Ari gali oh. All right. So, the gram-positive cell walls are dehydrated, no, and the pores of the cell are closed, no, causing the cell to shrink. So this one shows the gram-positive bacterium. Since wala siya yung lipid layer, ang diri upon addition of the alcohol, it will shrink. It will shrink. Pag nag-shrink, 'di ba, mag-close ang pores. So, halimbawa, eto may opening and then ay akong fingers. Okay. Paano ba?
No.
So, ganyan. Pag
Mas na-concentrate sila, ma'am.
Okay, nag-concentrate and the complex can no longer get out of the cell wall. Okay? So, nag-concentrate, nag-shrink ang cell, nag-concentrate further ang crystal violet iodine complex in the cell wall. So, it retained further the color, the purple color. So that's the reason why purple ang gram-positive bacterium. And after the addition of decolorizing agent, may mga washing stage na siya. You need to wash the alcohol, you need to, may mga washing. After sang crystal violet, may washing. After sang addition sang iodine, may washing. After addition sang decolorization agent, may mga washing stage na da. And then after that, you add the counter stain, the safranin. Okay. The color of the safranin is pink to red. Okay, red nga siya, red counter stain. So, ang effect na color, the color that's being manifested in the cells is pink to reddish purple. So, muna. So, what's the reason? I have several questions. What's the reason why gram-negative bacterium colors pink after the addition of safranin?
Ma'am, because kay nadula
Purple?
ang violet kag washing hangin kung introduce ang safranin as a counter stain, amo to na-retain niya yung pink color or red. Kaya wala nang stain sang crystal violet.
Okay, very good.
Kaya na-wash out siya.
Okay, very good. All right. So what is the purpose? Okay, thank you. Si ano to siya? Si
Icon, ma'am.
Roquero. Ay, teka. Hindi ko makita.
Si Icon.
Si Icon, okay. Roquero, ginman sila akong sulat. Okay, thank you, Icon. So, okay. What is the purpose of adding safranin? And why do we have to add safranin? Can we just stop sa decolorization stage during gram staining?
Hindi makita ang bacteria.
Yeah, that's the correct answer. No, it sounds so funny. Nga balon mo lang hindi makita ang bacteria, but amo ginaya ang reason why do we have to counter stain our bacterial cell. No, so this aids in the visualization of the bacteria, especially the gram negative. All right. So why is it that despite the addition of the safranin, the gram-positive bacterial cell or cell wall remains purple? Why? Nalobat ako.
Because ma'am, kay during the application of alcohol, nagsarado na yung mga pores ng cell, so hindi na maka-enter yung counter stain na safranin doon sa intrusion ng crystal violet.
Okay, very good. Another answer. Sorry, nag-charge ako ng aking laptop. Yes, thank you again, Icon. Ano pa yung may iba pa kayong answer?
I think, ma'am,
Practical lang yung answer sa question na yun, practical.
I think, ma'am, safranin cannot enter the cells na because it is already occupied by the addition of the crystal violet.
That could be possible, but well, maybe.
Ma'am, ang crystal violet mas darker sa ay sorry ma'am, magahod ni nampak ko lang misa.
Okay lang.
Ang
Sino ni siya?
Angel, ma'am.
Okay, Angel.
So, di ba ang crystal violet, ma'am, is darker than the red or like the safranin. So,
Okay.
hindi siya maka-color gid sa crystal violet.
Okay, very
Amo
good.
na nga marine color.
Ang iyang purple color. Okay. All of your answers are correct, no? Actually, gusto ko lang nga participate ikaw. That's why I am throwing some questions. But very good, no? And the answer to that is there is some sort of masking of color. Bali, mamask na some purple ang pink or reddish. Because as what Angel said, purple or violet is darker than the pink dye or the saffrony. Okay. Before we go to the next, actually this portion should be discussed by your laboratory teacher. Pero tagaan ka na kamo hint. Okay. Gin share ko na kadina how you should prepare your smear, but you also have to take note of the age of your inoculum or your cell or age of your bacteria that you're going to use in gram staining. Okay. So, ang ideal is around 18 to 24-hour old bacterium ang gamitin sa gram staining. What would be the effect if you use old cells? Kung old na bingang cell ang gamiton. Example, in the case of gram-positive bacterium, kung old cell ang gamiton mo sa gram staining, anong effect? Anybody would want to share?
More or less, ma'am, kung old cells, ma'am daw dead, mas damo in dead cells kaysa sa like alive. So like,
Alive. Okay.
so like, hindi siya gawa mag-work, ma'am.
Okay.
I think, ma'am, hindi na siya ma-stain since old cell, ang iya cells is ruptured na. Since
Okay.
ang staining requires
Intact.
absorbance ang stain. So,
Dahil lang kita. Class, when you answer, please mention first your first name. Para at least na-imagine ko kung sino akong ka-istorya. Kaya sa akong bala screen, wala ga-appear kung sino ang nag-answer. Okay? So, sino tong duwa ka nag-answer earlier? Ay, bago lang.
Angel,
Just a while ago. Angel and
Dinil, ma'am.
Daniel.
last name?
Laho, ma'am.
Angel Aquesta.
Ang listahan ko, the Angel, Laho, ganito. Last name ng iba, si Angel lang naka-first name din. Okay. What else? Amo ni tani ng mga types ng discussion na gusto ko. Nga daw nag-ask na lang kita mga questions. Mga practical nga situations na gina-ask. Okay. Ano pa ba ang reason? Sige, for this one, it's advisable to use 18 to 24-old cells because if you use old bacterial cells or old bacteria, especially if you're using a gram-positive bacterium, it will lead you to a false negative result. Now, false negative. Pag ginambal mo siya ng false negative, it is actually a positive, but it's actually a gram-positive bacterium, but it will manifest the characteristics of a gram-negative bacterium. Okay? So, what's the reason for that? Kasi syempre, nag-shrink na ang iyang cell wall because of old age. No? Tsakto to ang ginambal nyo earlier na it's possible na nag-shrink and nag-close ng mga pores and hindi na siya ma-stain anymore with the crystal violet and iodine, or hindi na maka-penetrate, or it can no longer absorb the dyes. Okay, so that's the reason. I have not encountered question on why what would be the effect if you use very young cells. Parang wala namang ganun. Kasi most likely, hindi ka man makakuha ng enough cells if you under culture your sample. Okay? Ano pa ba yung mga practical questions sa gram staining? Okay. For the decolorization step, there is a required number of seconds, no, that you will subject your smear, your bacteria, or you soak your bacteria sa ethyl alcohol. All right, it's around I think 15 seconds only, no? What would be the effect if you lengthen your decolorization step? If you go beyond the required recommended time.
Mapatay ang bacteria, ma'am.
Actually patay na ang bacteria sa fixation stage pa lang. 'Di ba gin-fix mo siya, paano mo siya to flame?
Yes, ma'am.
First name please.
Angel, sorry.
Okay. Oo. So 'di ba sa fixation pa lang ginamo mo na siya sa flame.
Yes, ma'am.
So medyo dead na to siya. So sa decolorization naman, why is it na, what would be the effect if you lengthen the decolorization?
Kasi ma-fully wipe out na gid bala ang ging fix mo na bala nga bacteria nga do hindi na gid siya visible kung i-examine mo.
Ma-wash. You mean ma-remove ang bacteria sa slide?
Ma'am, after ma-dissolve na.
Ma-dissolve ang bacteria mismo?
Yes, ma'am.
Okay. That would be true for gram negative. What happens sa gram positive if you lengthen the decolorization step?
Yung gram positive, Angel, ma'am. Angel, masabat.
Yes.
Basi yung gram positive, ma'am, like, kung ma-expose for more than 15 seconds sa decolorizer.
I am not sure ha. I-assume lang natin na 15 seconds gidman na. Pero hindi ako sure kung 15 lang gidman. Pero sige, let's say 15 for example.
Ma'am, basi ma-wipe out gid yung crystal violet kay gin babad mo gid siya sa decolorizer even though nga hindi siya gawa maapektuhan pero kay gin padugay mo gid basi madula ang crystal violet tapos ending yung magwa niya daw pareho sa gram negative nga na wash out yung crystal violet.
Okay, very good. That's the answer. No, it will again lead to a false negative result. Okay. Okay lang man niya sa sides ng gram negative because we are expecting it to be negative. But in the case, if your sample is a gram positive bacteria, if you go beyond the recommended time for decolorization step, it will result to a gram negative result. Okay, so the reason for that would be ma-leach out na ang complex that was initially formed. Okay, so ano pa ba ang pamangkot natin? So far, that's all for gram staining. And those things that I've mentioned in about gram staining are actually usually gonna ask the exam. Okay. Can you still remember what is the mordant that's being used in this technique? What is the mordant? Mordant. The mordant is...
Yes.
Yes?
Ang iodine?
First name?
Or no?
Name?
Natasha, ma'am.
Ano gani pala doon ni Natasha?
Britannia.
Okay, sorry. All of you, ano lang ako nasulat. Last name maging sulat ko, except lang niya kay Angel. Si Angel din. Kaya kita, ako mas lipotin yung last name kasi sa first name niyo. Okay, so the mordant is the iodine. So the purpose of the mordant is to fix the crystal violet dye sa cell wall. All right, so what is the counter stain? The counter stain is the safranin. Okay. Sige. So mga muna ang question sa exam. As I've said, parang ano na siya nga itong discussion, parang review na for the exam. And syempre, to elaborate on things that need to be discussed further, that needs further discussion. Alright, so amo ni siya mas namin is siyang representation of the of the gram stain technique, and it shows what will happen to the cell. So the chemical reaction in the cell wall here, upon addition of the crystal violet, it becomes purple, both becomes purple, but with the addition of the crystal with the grams iodine or the mordant, it will form the complex here, but not much, or has no effect or much effect sa gram negative bacterium. And upon the addition of the decolorizing agent, since wala siya nag-form some complex, it can easily go out, the dye can easily go out of the cell wall, plus na dissolve in aring layer, the lipid layer. And then with the addition of the counterstain safranin, it takes the color of the counterstain, the red dye. Okay? So siguro kung mag-quiz kita sa gram staining technique, perfect na ninyo. Okay. So what is this? Is it gram positive or gram negative? That one, parang bacillus siya, no? Is it gram positive or gram negative? Hello? Yes, Julian.
Gram positive, ma'am.
Okay, this is positive. And this one, of course, is negative. Okay, thank you, Julian. Ayan. So, we're done with the external structures, and ba, the 1 hour kita sa gram staining technique, no? Okay. So, actually, the rest naman ng mga topics sa slide ni are fully explained na in the slides. No, we don't have to go really in the details of this. Kaya daw, amo lang man to ang daw kinahanglan explanation. So, have you learned something new today?
Yes, ma'am.
That's good to hear. Actually, ako man, gusto ko man na we have this online class every week, because nagaka-paranoid man ako kung anong ginaubra sa mga estudyante ko during my class. Okay, if we do offline. Okay, so, okay, let's proceed. The first internal structure that we're going to discuss is the plasma membrane. No? Where's my plasma membrane? And it is composed of 2 layers of phospholipids. Now, briefly, this functions as this serves or facilitates the transfer of molecules, no? And it is where the oxidative phosphorylation happens. All right. So during oxidative phosphorylation, that's when there is breakdown of nutrients and production of energy. All right. So notice this one represents the plasma membrane, no? And this is the phospholipid bilayer. So this represents the head, and the heads are polar, no, or hydrophilic, meaning they like to stay in an aqueous environment. So we have the non-polar tails. These are the tails. We have the head, we have the tails. Nami ang ilang representation, no? Tapos siya in-interpret. Okay? And the tails are actually non-polar, or they want to be away from water, but they want to be in an oily environment. And since they are hydrophobic, they stays at the center. So ari di ang di ba may ara kata, we have a phospholipid bilayer. One set of the polar heads face the outside world, and this one or the outside aqueous environment, and this one faces the interior or the internal portion of the cell. Okay, so do you get it? Ano ba yung pwede kong
Yes, ma'am.
Parang wala talaga akong props dito. So, ayan. Teka. Wala ako props, guys. But I have this. So, this is the, phospholipid bilayer. So, ang ammonia nga portion is the head. This portion is also the head, no? These are the heads. The tails are inside, no? So, this portion, these heads here are facing the interior portion of the cell. This is the interior portion of the cell. And this one is the outside environment. So, ano man ang upod sang cell, other cells, and the water, and etc. This one ang sa insides of the cell, may mitochondria or whatever. So, muna siyang nag-face, no? Because we know naman for example, in a human body, we have our body has what? Is composed of water. And then the internal portion of the cell is also composed mostly of water. That's why very useful in siya nga ang the heads of the phospholipid bilayer are hydrophilic. Okay? May mga chat tayo. Let's check the chat. Okay. Okay. Noted ang inyong mga answers. All right. So maybe we can just skip this one. Mga membrane proteins, no? We have the peripheral membrane proteins. These are bound to the surface, this one. While the integral membrane proteins are bound sa sulod, no, embedded in the membrane. So ara sila sa sulod by one or more stretches of the wait. Ayan. So what are the functions of the cell membrane? As I've mentioned earlier, this facilitates the transport of the molecules and also the area where oxidative phosphorylation occurs. No, so take note that our cell membrane or the plasma membrane is a selectively permeable membrane. When you say selective, it has a selective permeability. It allows the entry and exit of some elements or molecules, while it also inhibits the entry or exit of other molecules. Ayan, may selectivity siya. So, pinapili niya lang. Right? So, we have examples of passive processes, the simple diffusion and the facilitated diffusion. So, maybe we can just skip this one. Are you familiar with the diffusion process? Familiar? So, simple diffusion, it does not require energy, and the movement of molecules is passive, no? No energy involved, no? And while the facilitated diffusion, it involves the participation of the transporter proteins or enzymes. Ayan, so this represents the transporter proteins. Okay, very detailed na ng slide, so we don't have to really discuss on this one. Is there any one of you who gets confused with osmosis? Kung ano ang movement sang water or sang water? Are you okay with osmosis? Naintindihan nyo na siya? Osmosis is defined as the diffusion of water molecules across the plasma membrane. So the environment surrounding cells may contain amounts of dissolved substances that are equal, less than, or greater than. So take note that it is not the solute that moves from one area to another, because the solutes cannot readily move No, it's the water molecule that's moving in or out or transfers from one. It's the water that transfers from one area to another, from one environment to another. All right. Mabulayan ako mag-illustrate sa inyo. Okay. So, this area has greater amount of solutes as compared with this one. Okay, black to pink or pink to black. So what would be the movement of the water? Is it from black to pink or pink to black? This one has higher solute concentration. This one has lower solute concentration. So what would be the movement of water?
From lower, Angel, ma'am. From lower to higher.
Okay, so ano siya? Lower to higher nga, solute concentration.
Yes, ma'am.
So are you suggesting
From
from pink to black?
to black. Yes, ma'am.
Okay, so that's correct. No, you always have to look for the environment which has the lower solute concentration. Pag lower ang solute concentration, meaning it is more diluted, no? And it has more water, so pwede siya usually ang tendency during osmosis is the movement of the water from the environment with lower solute concentration to the one with higher solute concentration. Okay, so that's it. What would be the effect of what would be the effect if the cell has higher solute concentration as compared with its environment?
Can you repeat the question, ma'am? Di ko
What will happen to the cell if the bacterial cell has higher solute concentration? This is the bacterial cell. Overexposed ang aking powder. If this bacterial cell has higher solute concentration as compared with its environment, what will happen to the cell?
This is Dinil, ma'am.
Yes.
Most
Ayan ang
probably,
name.
Laho, ma'am.
Okay.
Most probably, the water or the solvent will come out of the cell, causing for the cell to shrink.
And then the cell has higher solute concentration. Siya ang may damo solute.
Mas madako siya, ma'am.
Burst, ma'am.
Madako siya.
Okay. So tendency is it will absorb water, mag-transfer ang water from its environment into the cell. So ang tendency, ang cell is, the cell will maghabok, and eventually it will burst.
Yes, burst.
Yes.
Okay. Ang effect if this has higher solute concentration. What will happen naman if the cell has lower solute concentration as compared with its environment? Mas
Shrink siya, ma'am.
because it will
Mas-shrink. May loss of water, ma'am.
okay, some of its water will go out to the environment until the cell and its environment reaches equilibrium. Okay? Ma-reach nila ang isotonic condition. Okay, so that's for osmosis. Okay, you just read this: active transport, group translocation, etc. Okay, I think that's all for bacteria. No, medyo okay naman kita. Wait. I just want to discuss a little about ribosomes here. Class, when you check the PowerPoint, I recommend that you also make notes, no? Actually, for this lecture, I was able to make a parang one page back to back lang naman na notes. So you might as well do that para every time may ginabasa ka mo PowerPoint, may ara na ka mo notes, it will be easier for you to study later on. No, mas ano siya, para shorter na siya. Okay, for our first long exam, we have 4 modules to include to be included in the first long. So para mas malimit ang inyong basahon, dapat may notes ka mo. Okay. I wanna medyo may mga misconception about sa ribosomes eh. So ribosomes are or functions to decode the message in the messenger RNA, or ginabasa niya, gina-read niya ang messenger RNA, and this also helps in the synthesis of protein. So have you ever checked about S? Dere, di ba ang iyang unit is 70S. Have you ever wondered why 50 and 30S is called 70S? Tapos if you're going to look at the components here, it has 23S and 5S RNAs, and then it has 34 proteins. The other one has 21 proteins. Parang nag-wonder ba ka mo? Have you nakita nyo na siya, or you just note that, okay, if it looks like this, parang magkadaupang palad, ribosomes sila. Diba parang gano'n siya, no? Cute. Okay, take note that ribosomes has a small and large subunit. Smaller and larger subunit. So this is the large subunit. This one is the small subunit. Okay, have you ever wondered what an S there is? Okay, so S is the unit, no? And it stands for stands for Svedbergs. So Sverbergs, Svedbergs. Okay, this is the unit used to measure how fast the molecules move in a centrifuge. Okay, so parang ano siya, it does not refer to the 50 or 30 or to the number of proteins. It simply refers, the 70S here, simply refers the Svedberg units, no? That it takes during sedimentation. Pag in centrifuge, are you familiar with the centrifuge? Di ba pag nag-centrifuge, are you familiar with the centrifuge, guys?
Yes, ma'am.
Di ba pag nag-centrifuge ka, nagpa-settle ka sang solids? Right?
Yes, sa bottom, sang test tube.
Oo. So, di ba pag may solid ka sa test tube, i-centrifuge mo, mag-settle ang solid sa solid sa tube. So, muna ang ginobra to determine the Svedberg's value of the ribosomes. So, the reason why 50S na day kag 30S, no, is it takes 50 Svedbergs to sediment the larger chromosome, or the larger ribosome, and it also takes it takes 30 Svedbergs to sediment the smaller ribosome of a 70S ribosome na isang prokaryotic cell. However, if you combine the larger and the smaller chromosome, it gives you a 70S or 70 Svedbergs. That's why amo na siya 70 ang iya nga unit. But hindi na siya ma-explain if you're going to look at the number of proteins, the individual unit, subunit, hindi na siya ma-explain. So, iba ang pagkakadetermine ng 70S. Look at this eukaryotic 80S ribosome. It has a 60S larger subunit with 45 proteins, and it has 40S, no? Smaller subunit with 30 proteins, composed of 30 proteins. Ayan. So, iba man niya ang composition sang eukaryotic ribosome. But take note, no? Both the larger subunit and the smaller subunit of a ribosome contains proteins and rRNA, ribosomal RNA. All right. So, any question with the ribosome? Basta ipatrivia sa inyo. 50s plus 30s equals 80s. No, it's 70s. Okay. Okay, mga inclusions. These are reserve deposits, no? This is the inclusion. All right. So may function ang inclusion nga ni, no? The cells accumulate nutrients and use them when the environment are deficient. So mga ano siya, nutrients. And these are also macromolecules, no? Which are enclosed inside the cell to avoid increasing the osmotic pressure that would result in the dispersion of the cytoplasm. So it also has function in the maintenance of the isotonic environment inside the cell. Okay, modes of reproduction. So we have the asexual or binary fission and sexual by conjugation. So take note, during binary fission, bacteria duplicates its genetic material. So muna, no? The first stage is may duplication of the genetic material and then division. Nag-divide lang sila, that's it. There is a formation of the, anong tawag sini? Parang ano, partial formation of the cross wall. Cross wall ang tawag sini. Ayan. Okay. Excuse ako na class. May inom lang ako ng tubig. Okay, we have here a term generation time. So this is the time required for a cell to divide and its population to double. So it varies among organisms and environmental conditions. Now, most bacteria have a generation time of 1 to 3 hours. Others require more than 24 hours. So that's what the reason why very rapid ang growth ng bacteria, no? So for example, if you start with a single cell, after 3 hours, it becomes 2. And then after another 3 hours, it becomes 4. And then after another 3 hours, it becomes 16, no? 4 and then double. 4 times 4, 16. Tama ba? Tama, class? 12 double. Ay, 8 gali. So, and so on. No? So, we will have a separate discussion about generation time na sa module 4, I think. Okay, so this is bacterial conjugation. Notice, no, there is a sex pilus here that's involved in the process. So it involves a F plasmid, or this one involves the donor cell or F positive cell, and this one is the recipient cell, the F negative. The one with a sex pilus is the one that is the donor cell, or identified as a donor cell or F positive. So magko-contact yung pilus sa another cell, and then there will be activation and transfer of ayan. So plasmid is activated upon the attachment of the pilus, no? And then there will be ayan. So there will be an origin of the transfer sa plasmid. Magta-transfer siya dito sa donor or sa recipient cell until it forms its own plasmid molecule here. No? And then the complementary strands of both F plus plasmids are synthesized in the donor and recipient cell. So after the conjugation process, both cells becomes a donor cell, capable already of becoming a donor cell. Okay, we have the endospores, no, being used by microorganisms in their resting mode, no, nagko-convert sila into spores. Okay, and we have here the spore formation. Okay, maybe we will still pursue or push through with our alternative activity, no? Guys, are you familiar with tawag dito, with Google Classroom? Have you used Google Classroom in the past?
Yes, ma'am.
So pwede man gali kita ka Google
Yes, ma'am.
Classroom, no? Sige. Actually, the activity that I am proposing is, kasi I'm checking the time, it's already 2:44. I have a meeting at 3, no? The activity that I am proposing is, teka, let me check kung ano po yung next. Mga common na lang man ito, size and shape, arrangement, you can already, I am confident that you can study this part on your own na. Okay? So, di ba mga easy terms na lang? And the slide provided you with enough information, I believe. No? Okay. So, I think naubos talaga ng gram staining yung ating time. Ano ba yun? Okay. So, this is natapos na natin yung bacteria. We have not touched the fungi and the viruses. Okay. So, this is the activity that I am actually planning, no? Para makasave kita time and we can still push through with our computation discussion next week. I don't know if it's possible to do it sa Google Classroom, but pwede man siguro kita drive, Google Drive, no? I will post questions and then, bale, para siyang talk show na offline, no? I will assign some questions to, you randomly, and then I will ask you to, one of you also to critique or comment on the answer of your classmate. So parang parang hindi naman siya debate, pero parang nagre-respond ka doon sa comment. We'll do that offline. Oh, I'm getting choppy. Sorry. Okay. Hello. So okay na? So class, we will end with the first PowerPoint slide, no? We will continue the virus and fungi as well as the role of microorganisms to the ecosystem through an offline talk show type activity. So I will be assigning you as the one who will answer the question, and then there will be one who will critique or respond to the answer. So parang i-contest mo or whatever, whether you agree or disagree. Ikaw bahala. So parang you have two roles, no? First is to answer, and you will be asked to critique the answer of your classmates. Okay, so with that, we can really save time. Kasi after ng submission nyo ng Module 3 output, wala kayong gagawin ngayon. So that will be our alternative activity. So we don't have to spend much time on the discussion, online na discussion. Kasi I really want to allot our meeting next week for the computation part of the course. Okay? Clear ba? I have received a message.
Question, ma'am.
Yes, Angel.
Ang do alternative activity, ma'am, offline?
Bali,
Offline, di ba?
Bali, parang asynchronous, hindi kinanlan na mag-online kita, but you will access it online. Parang, siguro I will prepare a Google Doc na
Yes, ma'am.
editable by all of you, and then you just search kung diin ka to ma-answer, and then you also have to search kung in what item ka mag-critique or mag-comment back. So
Tanan, ma'am, nga, ako be, ma'am, ma-answer ko. Tapos, makritik man ko sa tanan nga answer sang classmates ko. Or mapili lang ko few people nga, ano.
Example, I will assign you to question number 1. So you answer that. I will put your name sa question.
Ah,
To be
okay,
answered
okay.
by Angel, okay? So question number 1 to be answered by Angel. To be critic by Feline. Na. So you answer in number 1, si Feline ang mag-critic sa number 1 mong answer. And then ikaw
Yes, ma'am.
will be assigned in number 10, for example, as critic. So you will only
Okay,
answer
ma'am.
1 question and you will only critic 1 question
One
also.
question.
1 answer.
Okay, ma'am.
Parang ganon. So,
Verified, ma'am.
yeah.
Thank you.
And that will already serve as your, parang ano na rin siya, reviewer nyo siya sa exam. And mas feeling ko mas dasig kita dira and then mas mag-learn ka mo. Okay, you can also participate in the research your answer. Okay? So are we good? May questions?
Yes, ma'am.
Sorry,
Okay, ma'am.
bago lang ko na-inform, I have a meeting at 3 gali. So I have to prepare for that. So thank you very much. I will expect your output within the day. And our day ends at 11:59 p.m. And I will also try to complete the questions, no? I need to prepare 17 questions for the 17 of you. And I hope to upload it within the day also. So nasa ano na siya siguro, guys, sa Google na Drive. Okay? Thank you very much. If you don't have questions anymore, we will end.
Thank
Wala
you,
na.
ma'am.
Bye. Ay,
Bye.
photo. Sige,
Hello.
sorry, picture taking. Picture taking. Those who are,
Wait lang.
who can open their video, please open your video. Naawa man ako.
Wait lang, ma'am. Di ko prepared. Charot.
Ay, makount na ko. Dahil lang. 1, ready na. 1, 2, 3. Liwaton ko to ha. Another one. Di na si Icon. Okay. Bye for now. Take care. Bye.
Bye, ma'am. Thank you.
Bye, ma'am. Thank you.
Thank you, man.